great and great. As I mentioned, I decided to do it very fast completely. We should start at the 8 o'clock, okay? As I mentioned, I changed the title of today's session because we have to have an experiment regarding to the measurement of the alkaline phosphatase, so it is not necessary to bring the lab coat, okay? So, in this session, I'm going to talk about the clinical enzyme. The clinical enzyme, I think in the previous semester you are familiar with the nature of the enzymes. Okay, they catalyze the reaction and they accelerate the rate of reaction. And some of them are clinically important. It means that they can consider as a symptom of the disease. And they also provide unique insight regarding some tissue damage, some organ damage, and cell damage. Why? Because we have some specific distribution of the enzyme. For example, we have a liver-specific enzyme, we have a heart-specific enzyme, muscle-specific enzyme, and if you remember, we have a different kind of enzymes. Some of them are extracellular, like an enzyme that participates in a blood clotting cascade, and also most of them present in the cells, inside the cells, and known as intracellular cells. So they entrap and hold by the cell membrane, okay? In a normal situation, we have a low concentration of this enzyme in the bloodstream. Sometimes some diseases, for example, some crash, okay, trauma, okay, any disorder can affect the cell membrane or the damage to the cells can result in an eruption and release of this enzyme in the serum. So these are very related to the disease. About the reference, you can refer to the chapter 29 of Titt's book and also Henry books. I will actually, if you have any problem, if you cannot understand the content of the slide, you can refer to the reference, and I put this chapter on the desktop, okay? So you can copy it if you have any problem for understanding some aspect, you can refer. But about your final exam, the content of your slide will be enough, okay? These are just for more information. What is the target of today's session? At first, I'm going to talk about some introduction about the enzyme, what are the functional enzyme, non-functional enzyme, and then I will introduce you some important plasma enzyme and how we can select the enzyme test, and actually the main part of my speech will be focused on a diagnostic serum enzyme, and I will introduce several kind of enzyme. As I mentioned, measurement of enzyme level in plasma indicates the tissue of the origin of the damage for diagnosis, prognosis, and assessment of the response to therapy. It means that when we say we can determine the tissue of origin, because I know that, for example, that tissue produces, for example, alkaline phosphatase. Heart tissue produces creatine kinase because they have a specific distribution, so you can guess which tissue is damaged according to the alteration in concentration of enzyme. About the history, if I want to talk about the history, at first, many years ago, more than 100 years ago, biochemists understand that alpha amylase activity in the urine is related to the acute inflammation of the pancreas. They followed the study of other enzymes, and then the use of serum as a diagnostic fluid for measuring enzyme activity starts at 10 years after this discovery, and they understand that another enzyme, for example, alkaline phosphatase, is specific and is very related to the liver disease. And 10 years later, the clinical value of acid phosphatase determined, and it's actually showed that there is a relation between activity of acid phosphatase in a bone metastasis from the some malignancies, especially from the prostate. As I mentioned, enzyme activity are the indicator, but very sensitive for the some cellular damage. It means that because, as I mentioned, very high concentration of enzyme are located inside the cells, and extremely small amount of enzyme can detect actually by their catalytic activities. So whenever you measure, for example, you take the blood from your patient and you understand that there is an increased activity of, for example, alkaline phosphatase, acid phosphatase in the plasma, it is a sensitive indicator. Maybe even a small injury, even a small cellular damage can result in an alteration in a normal range. For example, sometimes normal range is, for example, 30 to 100, and you measure the activity of enzyme and it is 300. So it means that there is some tissue damage. So by this way, you can determine some problem regarding to the damage to the liver cell or hepatocyte. And also we have some problem regarding to the liver cholestasis, inflammation of the pancreas, some disorder of the bone, and etc. So, do you remember in the previous semester I introduced in an enzyme session, I introduced two different types of enzyme, and we can classify them in two groups. One of them is a functional enzyme. What does it mean, functional plasma?
It means it's present in the plasma more than in the cell?
Yes. It means that they have a specific function in the plasma. Like, for example, blood clotting enzyme or lipoprotein lipase. They normally found in the serum. So, if you measure the activity of enzyme in the serum, normally they have activity, okay? So, they are considered as a functional. What about non-functional? As the name implies, they don't have any specific function in the serum, okay? What is the proper site to activate the activity inside the cells? Like, for example, any other enzyme. So, they don't have any function in the plasma. And mostly, for example, this enzyme mostly synthesized by the liver. But about non-functional, we have a wide distribution of this enzyme. Liver, muscle, heart, I don't know, pancreas, okay, or adipose tissue, okay. We have a wide distribution. So they increase, this is very important. So if you observe the increased activity of this enzyme, it means that we have some disease condition. So as a result of some tissue damage, like protein kinase, alanine, transaminase, etc. Because during the next slide, I have to introduce them some enzyme with a different form. I have to introduce a specific term. Have you ever heard the name of isoenzyme? Do you have any information? Who can describe what is it? Isoenzyme or isoform?
They are like two compounds that they have the same components, but they are arranged differently. Like for example, they both have say ammonia and carbon, etc. But
Yes.
in one of them, for example, the ammonia is in the middle, the other one's ammonia is at the end.
Yes, excellent. As your friend said, most of them actually have the same chemical structure and chemical formula because they derive from the same gene, okay? But what happened to them? For example, we have some post-translational modification. It means that we have a gene, they convert to the RNA and then they convert to the protein, okay? And we have some post-translational modification. What does it mean? For example, sometimes they give some amide group, sometimes they give a hydroxyl group or acetyl group, okay? The structure of the protein is same, okay? But sometimes they actually convert to another type of glycoproteins, okay? For example, alkaline phosphatase have a different isoenzyme or isoform. They have the same amino acid sequence, okay? okay? But what is the difference between the different type of, for example, we have a bone alkaline phosphatase, liver alkaline phosphatase. What is the difference? The content of the carbohydrates, okay? If you remember about from the previous semester, after translation, for example, maybe we have some glycoproteins, okay? Glycoprotein, what does it mean? What is the structure of the glycoprotein? Yes,
proteins,
carbohydrates and proteins. So when we add a carbohydrate to the structure of the protein, it's known as a post-translational modification. It means that at first I translate it to the protein, and after that I add, for example, glycoprotein. Okay, maybe one of them contains galactose, maybe another containing glucose. Okay, these are actually different type and known as an isoform. But actually, you should know that they catalyze the same chemical reaction. Okay, there is no difference in their reaction. All of them, for example, add a phosphate to the substrate, like protein kinase. Okay, they add a phosphate to the protein. Okay, but they have a different physical properties. Like, for example, they have electrophoretic mobility, they have a different kinetic parameters, and about localization. What does it mean? For example, some isoforms are present in the cytosol, some isoforms are present inside the mitochondria. Okay, these are about the localization of the isoenzyme. And also, we have some number. For example, lactate dehydrogenase is one of the best examples for the isoform because we have 5 types. As you can see here, if I put in the electrophoretic, actually in an electric field, and I start the electrophoresis, they move toward the cathode, okay? And as you can see here, to the anode, okay? The negative is the cathode and the positive is anode. And how we can give them the number, you should consider that this is the anode, again, this is the cathode. The most anodal isoform receive the lowest number, okay? So the isoform that is most nearest to the anode actually receive the lowest number, and actually the isoform that is near to the cathode receive the highest number. So also they have a specific organ tissue. For example, they can found in a liver. Some of them found in a muscle part. Okay, in the next slide I will describe. And about how we can select the enzyme test. For example, I think there is a problem to the, for example, liver. I think there is some problem to the RBC. Okay, which enzyme is the best choice to evaluate this damage? According to these charts, I'm going to show that the distribution of enzymes is very important. For example, this is a distribution of the alanine aminotransferase as the actually one of the most important clinically enzyme. As you can see here, this is a tissue distribution of the alanine aminotransferase. Okay, for example, and also this is a distribution of aspartate aminotransferase and protein kinase. For example, if I evaluate the tissue to the hepatocyte or to the liver, according to this distribution, which of these 3 enzymes is the best choice?
Alanine.
I think alanine aminotransferase.
Excellent. Why? Because we have the highest concentration. So, for example, if I want to evaluate, for example, anemia. In anemia, the enzyme that present inside the RBC release in the serum. Okay? So, if I want to evaluate the damage to the RBC or erythrocyte, alanine aminotransferase is not a good choice because maybe I make a mistake with the liver disease. So, according to this chart, the tissue distribution can be considered as a selection of the enzyme test. And in the next slide, I will show, for example, which slides, which enzyme is specific to which organ. Have you ever seen the lab report about the enzyme? Can I write here?
Yes.
For example, we have an X enzyme, and the normal range is about, for example, 33 to 100 international unit. For example, this is the unit of enzyme. As you can see here, the reference interval or normal range is not start from 0 or is not a 0. Why? And if you actually observe this distribution and this activity of enzyme in the serum is considered as a normal. It is not related to any pathological situation. What do you think? Why we have a little amount of enzyme in the bloodstream?
So that it can perform its functions. And if we don't have any of the blood, level 0, then that means it's not doing its function. So that's that would be a pathological issue.
Yes, it's also correct. But maybe it's better I change the question. What is the source of this normal range? What is the source?
Is this the amount that is needed to perform the function without being too little?
No, you should consider this point. For example, these are the normal range of the non-functional enzyme. I mean that in the bloodstream or in the serum, they don't have any specific activity. But why, according to this slide, you can guess, why we have a little amount of this enzyme in the serum? Why?
Okay. According to the slide, because when cells die, they release their enzymes into the blood.
Excellent.
So if there's too little, it means the cells didn't have that much enzyme, and if there's too much, it means the cells are dying too quickly.
Yes. So we have a hectic life. We have a normal cell turnover. It means that whenever you have a normal range or reference interval and it is not 0, as your friend said, this is as a result of normal cell turnover. So we don't have 0 normal range, okay? But other than, for example, if you have a 300 international unit activity of your enzyme, it means that there is some damage, there is some problem. Maybe this can result in MI. Maybe it can result from the cancer. Actually, for example, according to the textbook, we have some factors that can affect the integrity of the cell membrane, like hypoxia. Hypoxia, it means that we have a low oxygen supplement, and also maybe some oxidant, some toxin, and some trauma, okay? All of these factors can affect the integrity of the cell membrane and result in a high concentration of the enzyme, and so it is related to some pathological. But it is a physiological normal range, yes.
But how do we know that if we have too much of the enzyme in the blood, it means that the cells are dying too quickly? What if the cells just have a very large amount of enzyme in them, their concentrations increase?
Actually, in the next slide, I will describe what all the time, if you have a higher, high activity of the enzyme, you can take an off, there is a problem, okay? But we have some guideline according to some specific enzyme. For example, in some specific enzyme, if you have 2 times upper than normal range, for example, is related to some acute disorder. 5 times is related to some chronic disorder. But it's not actually correct to all the enzyme. Each enzyme has a specific guideline to determine what is the problem. In the next slide, I will describe. Okay, these are the most important diagnostic serum enzyme. As I mentioned, we have a tissue distribution of the enzyme. For example, we have some specific enzyme, and you should know for your final exam. Maybe I ask you, so this slide is very important for your final exam. You should know that what are the specific enzyme for the muscle. You should know that creatine kinase and aldolase are the specific enzyme for the muscle. In the next slide, I will describe one by one. About the liver enzyme, because liver considered as a factory of the body, so it contains a different type of enzyme and they have a different role, especially during the metabolism, like aminotransferase, glutamate dehydrogenase, alkaline phosphatase, and other enzymes. Some of them are that are of clinically important. So in the next slide, I will focus not about all of them, okay? Because, for example, about 5-nucleotidase is a symptom of some specific disease. So you don't suppose to know the details of this enzyme, but you should know the name of the enzyme that is specific to the liver. In the next slide, I will introduce some details about some specific enzyme. And for example, about pancreatic enzyme. In a pancreas, what is the function of pancreas?
Secretes insulin and glucagon.
And also, it's a very important organ. And you know, malignancy of this organ can actually result in a death less than 1 or 3 months. Okay, why? Because the function of this organ is very important to the body. As your friend said, because this is the site of secretion of many hormones, especially insulin, controlling the fasting blood sugars. Not only they produce, this organ produces some specific hormone, but also it can also produce some enzymes that are responsible for the digestion of the food. Okay? For example, for the digestion of the lipid, we have a lipase. For the digestion of the carbohydrates, we have a amylase, and also we have a trypsin for digestion of the protein. It means that if you have a problem about the pancreas, so you cannot digest your food source, okay? And also we have some bone enzyme. For example, alkaline phosphatase and acid phosphatase. And each of them are specific. For example, do you know we have 2 specific cells in the bone? One of them participates in a bone formation, another one for resorption. Osteoblast and osteoclast. Osteoblast produce a bone, they form a bone. So if you have some, for example, in a normally growing children, okay, in normally growing children, the activity of the osteoblast is high. It's normal, it's not related to the any pathological disorder, okay? So if I measure, for example, inside the osteoblast, we have the alkaline phosphatase, and in the next session I will have some experiments regarding to the measurement of the alkaline phosphatase, okay? Alkaline phosphatase can find actually in a liver, and also in a bone, and also in a placenta, okay? For example, because as I mentioned, this enzyme can be found in the osteoblast. So in a normally growing children, if you measure the activity of enzyme is normally higher than normal range. So it is not related to any pathological or bone disorder. So as a physician, if you actually only have a result and you don't have any information about the age of your patient, maybe you think that your patient has some problem about the bone, okay? For example, if you have information about the age or demographic information about your patient, if you know, for example, he or she is 12 or 11 years old, so you can interpret as a normal result. But for example, if your patient is 50 years old, 55 years old, it's actually, it can related to any problem regarding to the, I don't know, osteoporosis or any problem regarding to the bone. So you should know the information of the patient, because sometimes age, gender, race, okay? For example, sometimes pregnancy can affect the normal range. So you should have this information to interpret your result. And also, we have the acid phosphatase, and this enzyme is specific for the osteoclast. Okay, so whenever you have some resorption of your bone, the activity of this enzyme is high. And also we have a miscellaneous or other enzyme, lactate dehydrogenase and cholinesterase. These enzymes are for more information. And in a specific situation, physician order to measurement activity of this enzyme. But for normal evaluation and normal checkup, we'll have another enzyme, and some of these enzymes are important. Okay, I will start with the muscle enzyme. As I mentioned, creatine kinase and aldolase are muscle-specific enzymes. As the name implies, what is the kinase? Excellent. Whenever you have a name of kinase, it means that this enzyme add the phosphorus from which source? What is the
ATP.
source of? Yes, excellent. ATP all the time. So, as your friend said, kinase all the time add a phosphorus to the substrate. And what is the source of this phosphate? ATP. Okay? So, as the name implies, this enzyme catalyzes the reversible, as you can see here, this is a reversible reaction, reversible phosphorylation of the creatine kinase by the ATP. When the muscle contracts, okay, when the muscle contracts, what happens? ATP convert to the ADP. Okay? When the muscle contracts, for example, I take it, okay? My muscle requires some energy to take this control, okay? So, my body actually uses energy. And what is the source of energy? ATP. So, because as my muscles contract, ATP, as you can see here, convert to the ADP, okay? And is it actually a high energy compound? ADP is high energy or no? Which of them? Which of them is more energy? ATP. So, because our body wants to regenerate and reform ATP, it can use a phosphorylated form of creatine. So, in a muscle, we have a phosphorylated form of creatine as a reservoir of the energy. Okay? So, whenever my muscle uses the ATP, it produces the ADP. Because actually our body wants to produce more ATP for another contraction, it can detach the phosphorus from the creatine phosphate and bind to the ADP and again produce the ATP. It means that normally, as you can see here, we have a different pH for the forward and reverse reaction. At neutral pH, like in muscles, the formation of the ATP is favored because our body wants to regenerate the source of energy, or ATP. Is it clear? So, we have a creatine phosphate. What is the role of creatine phosphate? Help and donate phosphorus to the ADP to produce ATP as a source of energy. So it's considered as a high energy compound. What is the structure? About the structure, creatine kinase is a dimer. It means that we have two subunits. One of them is B, another one is M. B is resemble brain, M is muscle. So about tissue distribution, in the next slide I will show. But what do you think about which of them frequently found in a brain?
Brain.
B, yes. Which of them frequently found in a muscle? M. Okay? So these are the different forms, so we have a different combination. If we have a BB, it's known as a creatine kinase 1. MB is 2 and MN is 3. As a physician, you have to memorize because you have to interpret the result, and some laboratory just write the report creatine kinase 2. So you should know that which of them is actually MB, which of them is BB, and which of them is MM. These are very, very important for your next years because you have to interpret the result of your patient according to this result. And also we have another kind. This is a mitochondrial creatine kinase and is located between the inner and outer membrane of the mitochondria. And also we have another type, macrotype, maybe in a specific situation, in a rare situation, some laboratory report this result. And macro, it means that this is it.
What is the location of MV?
In the next slide, I will, it's actually tissue distribution in the muscles, okay? Especially in a tissue, organ, like a leg, like a hand, okay? In the next slide, I will show the specific tissue distribution. And about the macro, it means that this is a combination, sometimes BB with the immunoglobulin and sometimes AB with the immunoglobulin. And these are not of pathological significance. And sometimes we have some oligomeric form of this creatine kinase. And about the macro, we have, for more information, we have a type 1 and type 2. If you have some report about type 2, it's related to the hospitalized patient, a patient who are severely ill, okay? If you measure the activity of macrocreatine kinase type 2, they are normal, they are not normal, these are higher than normal. And you should consider at this point, the normal range of this enzyme can affect by the sex, age, race, and muscle mass. What does it mean? It means that yes, men and women have a different muscle mass. So the normal range of this enzyme is different between men and women. So for interpretation, you should consider this point. Okay, it's very related to the tone and also mass of the muscles. Okay, for example, muscular athlete, because they have a large amount of muscle, they normally have a higher, sometimes higher than normal. And for example, if you have a severe exercise, okay, and you have a vigorous exercise, normally, you have a maybe higher than normal range of this enzyme. Okay, so we should consider and you should ask from your patient, are you a professional athlete? Okay, if he or she is professional, you can interpret as a normal. Okay, you should consider all these points. Oligomeric actually is a combination of the, for example, 3 or 5 creatine kinase that actually mitochondrial form. When they combine together, they form a large complex and consider as an oligomer. And this is not related to this point, okay? Oligomer introduce a specific kind of creatine kinase, but these are not actually of pathological significance. These 3 types is important for interpretation of your result. About tissue distribution, as you can see here,